Hello and welcome to Nikolai's genetics lessons, and in this video I'm going to talk about primers, and here's a problem. The following is a typical target sequence of DNA at an STR site. STR stands for the short tandem repeats. You will need to identify the repeated sequence in the upper strand. It is composed of 4 nucleotides. This repeat does not start at the far left of the upper strand. The primer binding region at the upper left is where the primer will bind. The repeat is to the right of that primer. Note that the underlined primers are different for the upper and lower strand. So let's analyze what we have here. This is upper strand, this is lower strand, and underlined sequence on the upper and lower strand is a site to which we have to design primers in order to make a copy of this fragment. One more time, STR stands for the short tandem repeats. This is core sequence that will be repeated multiple times. For example, CTG, 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 or CTT, CTT, CTT. Such STR core sequence can be different. For example, it can be 3 nucleotides, so it's going to be a repeat of 3 nucleotides over and over again. It can be 4 nucleotides and so on. Our first question: what is the sequence of the 4 nucleotides repeat in the upper strand? Let's analyze. This is upper strand, and we do not take into account this part, because this is going to be where primer is going to bind. So we have to analyze this part of the sequence. And right away I see 4 nucleotide repeat, which is TTCA, 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 and that's it. We don't have any more repeats. So the sequence is T, T, C, A. This is going to be an answer to the first question. And next question: how many repeats are there in the upper strand? Again, we have 1, 2, 3, 4, 5 repeats. And by the way, that also means that in the lower strand we have the same repeat, but different nucleotides going to be, but still we are going to have 5 repeats. But because of the complementarity, here we are going to have a repeat of AAGT, AAGT, and so on. Now let's move to the question C. What is the sequence of the forward primer binds to the upper left sequence, which is underlined here? In order to make copy of this fragment, we have to raise the temperature. So the temperature, about 100 degrees of Celsius, are going to break hydrogen bonding between complementary nucleotides. As you see, thymine is base pair with adenine, cytosine with guanine, and so on. And they make hydrogen bonding between them, which we can break chemically or with a temperature. When performing PCR, we use temperature to separate strands. So as you see, now strands are separated, and we need to build another complementary strand for this strand and for this. So we are going to start with 1 fragment of the double-stranded DNA and end up with 2 fragments, identical fragments of the DNA, double-stranded DNA. So we will make a copy. And we told that this is going to be a primer binding site, and because these 2 strands are complementary to each other, we instantly know the sequence of the primer. It has to be the same, so the same as we see in this strand. Or we can say adenine base pair with thymine, thymine with adenine, cytosine with guanine, and guanine with cytosine. As you see, the sequence is the same. Now, what about primer ends? For example, what's going to be primer end here and here? Polymerase only can add new nucleotides from 5' end to 3' end. So we instantly know that here have to be 5' end, here have to be 3' end in order to build a new strand in this direction. So again, here is going to be 3' end. So we start with 5' end, and new strand of the DNA, complementary DNA to this one, would grow in 3' end. That means that we instantly know that this is going to be 3' end here and 5' end here, because these two strands of the DNA are going to go in different directions. Because this is going to be a complementary strand to this one, we also instantly know that here we are going to have 5' end and 3' end here. Again, the ends here are the same as in primer. By the way, we can give an answer to the question C. What is the sequence of the forward primer? And it is 5' end, T, A, G, C, and 3' end. Now let's talk about the sequence of the reverse primer. This is question D. This is primer binding site, and as you already know, new strand of the DNA grows from 5' end to 3' end. So we have 5' end here, 3' end here for our primer, and the sequence is going to be the same as we see here, because these two strands are complementary. So let me underline it. Now check it out. Adenine base pair with thymine, thymine with adenine, guanine with cytosine, cytosine with guanine, cytosine with guanine, adenine with thymine. We got the same sequence as in complementary strand, and this is a sequence of the primer, and directionality is the same as we see here. From 5' end to 3' end, new strands are going to grow in this direction. So now you see from one double-stranded DNA, we got two double-stranded fragments of the DNA. And the sequence of the reverse primer is going to be 5' end, T, A, C, G, G, T, and 3' end. Why I didn't give an answer 3' end and the sequence 5' end? Because when we place an order for primers, it doesn't matter if it is forward primer or if it is reverse primer, we always give sequence from 5' end to 3' end. So this is the sequence for the reverse primer from 5' end to 3' end. And this is all for today. Subscribe and see you in the next video. Goodbye.